Hi, everyone. Welcome to the next episode of Editors in Conversation in Bio Edition, a show about the tiny organisms shaping our world and the scientists that are uncovering their stories. I'm your host, Marvin Whiteley. I'm the editor-in-chief of InBio, and today we're going to be diving into the fascinating world of bacteriophages, or phages. Before we begin, I should point it out that today's recording spans almost half the planet. It's late afternoon here in Atlanta on a Monday, while our guests are in Australia and they're joining us bright and early. I think it's Tuesday morning there. So our guests today, they're literally speaking to us from the future, and I think which is kind of cool because today we're discussing a phage and bacteriophage and some really cool findings that were recently published in InBio. So I'm delighted today to be joined by Trevor Lithgow and Sabrina Suhani, two of the authors of this study. So welcome to both of you.
Thanks very much, Marvin.
To get us started, I'd love for each of you to briefly introduce yourself, tell us your role in the study, and a little about maybe your broader research focus. So Sabrina, let's start with you.
Sure. Thanks, Marvin. So I'm Sabrina. I'm a third-year PhD student in Trevor Lithgow lab. So basically, I work with Staphylococcus bacteriophage. I isolated this cool phage in 2023, which we found out that it's a contractile phage, but at the same time, it's very flexible as well, which is kind of cool. And we sort of conducted our studies towards characterizing this phage, the functionality and the flexibility of this phage. And we could see that, it's kind of cool that it has a range of flexibility, and which we have talked in our paper that it helps in infecting the Staphylococcus cells as well. So that's sort of about me and my studies.
Cool. Very cool. Trevor?
Yeah, so I'm Sabrina's PhD supervisor. I'm a professor in the Department of Microbiology in the Monash University Biomolecular Discovery Institute. I've been at Monash for almost 15 years now, and the study that Sabrina took from idea on the page through to this paper in InBio has been really exciting, and several people in the lab have sort of contributed towards the final stages of this paper.
Yeah, cool. Well, that's how it is normally now, right? The studies are getting bigger and bigger, and you need help from lab mates a lot of times to finish up. So before we get in and really talk about the specifics of the study, which I think the study is super cool, it's why I picked it. As soon as the paper came out, I thought we definitely have to do a podcast on this. I think I'd like to take a step back for a moment, and maybe Sabrina, just for listeners who may not know or think about phages all the time, can you just tell us exactly what a bacteriophage is?
A very cool question, but a very basic one. So I'd like to take a step back and would like to literally have the introduction that I have once I started my PhD, which is a bacteria eater virus. So we know that the bacteria that attacks our body or creates these diseases in our body, we can treat them with antibiotics, but they are smarter than humans, so they become very resistant towards these antibiotics soon. So we have these cool viruses in nature which can basically attack the bacteria, kill the bacteria, but at the same time, they don't harm the human body. So we can take advantages of these tiny little viruses against various disease-causing or antimicrobial resistance bacteria to sort of kill them, but at the same time, preserve the natural human microbiota.
Yeah, so viruses that eat bacteria is kind of what you said at the beginning. So that makes a lot of sense to me. I think I read something. We work a little bit on phages, and I've read, that people say that they're the most abundant biological entities on Earth. Have you heard this? Is this true?
Yeah, that's kind of true.
And, when I think about phages, right, I think about a phage, a virus moving around. They don't have their own motility, right? They're kind of just moving around. And if they kind of bump into a bacterium that they could infect, Trevor, could you go through, what the stages, what are the stages of a phage infection?
Yeah, sure. So, and in fact, this is, I guess, the main motivator in the lab for why we like working on bacteriophage is that we are really interested in this encounter between the initial encounter between the virus and the bacterium that it's about to infect. So the very first step would be, as you said, oftentimes, so the bacteria we work on, Klebsiella, is itself also immotile. And for the staph that that Sabrina's project is focused on, likewise. So it's, two entities that are tumbling around in a medium, and it will be a chance encounter when the phage first strikes the surface of the bacterium. And so we're quite interested in this first encounter where there has to be some sort of receptor on the surface of the bacterium, and there has to be some sort of receptor binding activity on the virus itself, on the virus particle, that would mediate an interaction that would be strong enough to give the phage the small window of opportunity that it needs in order to get itself organized and inject its DNA into the cytoplasm of the bacterium. So it's oftentimes the case that in order to find the receptor, or indeed to get to a second receptor, this would require some sort of enzymatic cleavage event at the surface of the bacterium. And that's certainly the case in the case of the Bacteriophage JS1 that Sabrina worked on.
And for staph, are the receptors mainly proteins, or are they also polysaccharides? What are the main? Yeah,
we think both.
Okay.
So we in the case of, so I should say, just to be completely clear, in this paper we didn't determine the specific receptor that was responsible for the interaction with JS1, but it would be a reasonable working hypothesis that it would be the cell wall peptidoglycan, so the actual carbohydrate that the bacteriophage is interacting with in the first instance. And what Sabrina could show was that there are enzymes on the surface of the virus particles that can degrade and perhaps help to sort of edge the phage closer to the surface membrane, which ultimately is where it has to get to in order that it can inject its DNA into the cytoplasm.
Yeah. And I think this sort of takes us, we've mentioned that Staphylococcus is the host for these, for this phage. And, when I think about Staphylococcus, I think probably most listeners, they think about Staph aureus, right? They immediately think about Staphylococcus aureus, MRSA, methicillin resistant Staph aureus. But, you guys like focused on other Staphylococci, and then, and we don't really talk as much about them. What was the motivation, I guess, Trevor, for looking at these other Staphylococci?
Yeah. So I would say the main motivator was Sabrina. So when Sabrina came to my lab, she came with an intent to work on Staphylococcus. Initially, we talked about working on Staph aureus, exactly as you said, that, it's the most famous one, it's the most obvious one, it's also the one that's most likely to appeal to a reviewer of your paper or of your PhD thesis, just because it's such an important pathogen of humans. But we, in the lab, we don't tend to go to clinical wastewater in order to isolate either bacteria or bacteriophages. We tend to go a little bit more off the beaten track. And so Sabrina went to a community wastewater. Well, she did not go to the community wastewater plant, but she accessed water from the community wastewater plant. And then played it out for Staphylococcus without bias. So it wasn't that we were aiming to get things that were not aureus, but the plating work that she did, and I'll let her chime in a moment just to describe what she found, but the plating work showed us that the most prevalent staph that you could plate out of that water were not aureus, and in fact they were multiple other species.
Yeah. Yeah. I mean, I think so, Trevor mentioned wastewater. So Sabrina, that's where we have to start, right? It seems like when I talk to phage people, they love wastewater, right? Everyone talks about using wastewater, right? So why wastewater? Why is it such a powerful place to actually go out and, and identify phage? Why is everyone using that?
Well, I wouldn't say that for all the phages, wastewater is the ideal collection points, because in my lab, my other colleagues are isolating phages from like river water and some running water as well. So I think in the case of staph, mostly because I think it's a skin disease, like mostly the staph bacteria causes the skin diseases. And I think that with the with the water that goes from the hospital or from the industries that goes to the wastewater, it is a good source of the bacteria as well as the virus as well. In our case, most of the bacteria that we had, like Staphylococcus xylucis or Staphylococcus lentis or some other strains, they are mostly, let's say that they're not exactly clinical pathogenic strains, but they can be used in meat fermentation and some other cases as well, which makes sense that they go to the wastewater. But in our case, we could ideally extract a phage from the wastewater that could actually infect clinical MRSA as well. So it's kind of cool.
So how do you do this? So you went to the wastewater and you get a sample, or someone gives you a sample, I guess, is what's the first step? How do you do this? Right? I don't want people to do this at home necessarily, but if they were to do it at home, what would be the way you would do this?
Well, I'm glad that you asked, because Trevor knows that I almost spent the first 6 months of my PhD just to get with the legality of like getting the wastewater. How do we establish it in our lab? And we were sort of fortunate enough that another department, like some sort of engineering department in Monash University, they used to collect the wastewater for some of their researches as well. And they were kind enough to let us use those wastewater from the collection that they sourced the wastewater. So ideally, I have collected, let's say, 21 liters of wastewater from them and used them to extract 16 bacterial species and one bacteriophage named as JS1 as well.
Okay. Oh, well, that's cool. So I guess, Trevor, one of the things that, when I read this paper that was really surprising to me, I've worked a little bit on Pseudomonas phage, right? And I know a little bit about Pseudomonas phage, but the phage that, that we work with are certainly specific to a certain species, right? And sometimes even a strain, right? But this phage that you found, I mean, it seems like it infects a lot of staphylococci. I mean, how odd is that? And was that surprising to you?
Yeah. So it was certainly surprising to me. So we hadn't, like I said, this was our first venture into the world of Staphylococcus and Staphylococcus phage. So most of the work that we do in the lab is primarily it's with phage that infect Klebsiella, and that's exactly as you said, Marvin. So most of our phage are specific for a single species of Klebsiella, and indeed for a particular strain. It's normally dictated by the capsule type of the Klebsiella, so the polysaccharides on the surface. And it's very rare that we find a phage that can infect two different capsule types. It happens from time to time, but it's rare. So the fact that this phage JS1 was so gregarious in the way in which it would just sample, different species of Staphylococcus was surprising to us. It turns out, I don't know if it's a general trait of staph phages or not, but it turns out that our colleagues over in Western Australia had isolated a couple of other phage that they then provided to Sabrina to do the same testing side by side with her JS1. And although they're not quite as, capable in terms of infecting diverse species and strains of staph, that they too, they're not species specific. And so I feel like this might be something that's perhaps common with staph phages, though it's probably too early, certainly too early in terms of what we've seen or read about to make that comment.
Oh, yeah, that's pretty cool. So, one of the things, obviously, that one of the cool parts of the paper are the images, right, that you see, right, of the phage, right, and this sort of curved tail that you actually see. And so were you the first one to see that, Sabrina?
Well, yes and no. I mean, I am technically the first one because I work with my collaborators and I got the images first. But yeah, I shared them with Trevor and my lab mates immediately, like in the lab meeting or even personally as well. So it's sort of at the same time that we all were surprised together.
Well, but you saw it before Trevor, so that was good, right?
Yes.
Technically, yes, because I work with the samples. So yeah.
Yeah, I was wondering, what you kind of thought, right, as a student, right? I remember when I was a grad student and I would get some data that was kind of unique or something you didn't expect. And I would always sit there and think, could I have screwed this up, right? Like before I would actually show my advisor. Did you actually think that, or did you just run in and show the pictures?
Yes and no, both. Firstly, I actually didn't have that much idea that the contractile phages were not as flexible as JS1 as well. So initially, I thought it's something normal, but then when I was reading the papers, I got to know, oh no, it's not the same, and maybe I have screwed something up. And I think we, as an entire lab, thought the same, that it's an artifact of the electron microscopy, or it's some sort of sample preparation that I have done. So I've done it at least 8 or 9 times with different phage preparations to be sure that I haven't screwed anything up and it's not an artifact of the electron microscopy, and it is a fact. But as you said, Trevor can tell it better, that we ourselves had a hard time to believe or even hypothesize what's happening with the morphology of these phages. So I'll let Trevor talk about it.
And maybe Trevor expand a little bit and let folks know what a tail does in a bacteriophage.
Yeah. So it was exactly as Sabrina described, and I remember the lab meeting when she had presented. So, we like to do biochemistry with our microbiology. And so she had, at the same time as presenting the images that showed the curvature in the tail, she also had the mass spectrometry to say that it had a base plate and it had a tail sheath and so on. And these are all features of a contractile phage. And I do remember the discussion, which, says something about the people in the lab. It was all very courteous and respectful, but it was certainly argy-bargy in terms of, but it's contractile, so it can't be flexible. Oh, but it's bent, so it can't be contractile. And then in the end, settling down to say, well, turns out it does both things. And so I think that was a surprise. And it was also one of those nice things that happens in a lab from time to time where you realize that it's okay to be surprised and it's okay to find something that, you didn't expect to find. And then you can get really excited about it and try and work out what it means. The tail itself on a bacteriophage, it does a couple of things, but the major function of the tail is to allow passage of the DNA, which is bundled up at really high density in the head, the capsid of the bacteriophage. And the tail forms a conduit so that the DNA can rush out in a linear fashion once the membrane of the cell's been breached, so that the DNA from the phage can enter into the cytoplasm where it can be replicated and transcribed and lead to the production of new progeny. A contractile phage gets it's the most easy to understand mechanism by which the tail can do this job, because once it settles onto the surface of the bacterium, the contraction of the sheath will force the tail tube that's inside the sheath through the membrane and into the cytoplasm. And in the case of a gram-positive, that means direct entry into the cytoplasm. And during that contraction, what also happens is that the end of the tube opens up so that the DNA can literally rush out through the tube. In non-contractile phage, and the non-contractile phage are the ones that you normally see with curvature, and we assume flexibility, though no one's shown that these things are flexible. Those non-contractile phage, it's probably much more interesting in terms of how the DNA gets through the membrane, because you don't have this forceful contraction event that can drive the tail tube through the membrane.
So, so essentially what you have is a tail and it kind of squishes, right? And when it squishes, right, it pushes the DNA, provides the energy to push the DNA through. But then, the really cool part of this paper is, well, how do you do that through a curve, right? Like you're curved, right? And now you're going to squish, right? And then, and so, I don't know. Sabrina, tell us some thoughts on that. How's that working?
Well, so we have sort of tried to have a hypothesis in our paper that from some of our SCM scanning electron microscopy images that we did to try to understand how it's infecting the host. And we could see in some of the cool images of our paper, which I remember is figure 6. So we could see that when they're infecting the cells...
Trevor doesn't know what figure it was.
No, he knows. I think that's why he's laughing, because we have been through this figure so many times that I do remember them now. But so we have, I think it was cool to see that even though when they were infecting the bacterial cells, they were sort of bent as well. So we kind of expected that they are bent on themselves when they're not doing nothing and they're just laying on the bed and they're bent and they're flexible. But when they're doing real work, like infecting the cells, they might be straight and they might be just having the contractile tails. But we could see from the images that they are sort of have like using this flexibility to go through the cells as well. Because if you think of the size of the cell wall, which is like sort of 44 nanometer thick, and the tail of my phage is approximately 220 nanometer. So if you think of that long particle to go inside a cell, as we have hypothesized in our paper, that it sort of bends and it gives the flexibility to go inside the cells, and then it can contract and inject its DNA into the cells. So I think it's kind of cool.
So when you're thinking about like for gram positives, right, just for folks, with the peptidoglycan layer, it's kind of like a mesh work, right? And it's got holes in it. They're like 27, 28 kilodaltons or something. There's holes, right? But as a phage, you either have to digest your way through, is that right? Or you have to kind of meander your way through, right? Is that right? To get to, is that what you're thinking?
I think it's both. I'll just jump in, Sabrina, but then jump in afterwards. I think it's both things, Marvin. So there's been in the not too distant past, there was some really nice work done with atomic force microscopy on staph cell walls. So it's clear that, unlike the textbook cartoons that show that the cell wall is, big, thick, and kind of impenetrable looking to a phage, actually there are all sorts of canyons and valleys and holes that, permeate through the wall. But again, coming back to what we discussed before, a phage doesn't get a chance to see that, and it certainly can't move towards an easy access point. And so wherever it happens to strike the wall, sometimes it might be quite far into the wall, other times it might be right up on the top, and then it would be helpful, and we think this is the case, to have enzymes on board that can help you degrade through the cell wall. We imagine, but again, I don't know what experiment we could possibly do to test this, but we imagine that having flexibility in the tail would provide for this meandering possibility. So if there are pre-existing canyons, or if you just enzymatically engineered one for yourself, that you would be able to reconnoiter through with a flexible tail more readily than if you had a straight, rigid tail. But we don't know, and of course, evolution doesn't work that way, right? It's not that you get to evolve something because it'd be really handy. Right. It's just that they happen to have these properties, and it may be useful to them.
Yeah. Well, I guess when I was thinking about it, so when you, to get to these sort of ideas, I mean, you guys did some structural modeling too, right? Like to try to understand how does that work, Sabrina, and what did that tell us about it?
So yes, initially we didn't think about the structural work, but then we sort of had the idea, Trevor mainly had the idea that if we could predict the structure of the tail tube, that might tell us something. What is it different from, like, let's say it sits in between the non-contractile phage and the contractile straight tail phage that we previously used to call like myo-like or sipho-like phages. So we tried to sort of have a comparison of JS1 tail tube with one traditional non-contractile and one traditional contractile phages tail tube. And what we could see from that comparison is that the tail tube of JS1 is missing some of the beta strands and some of the beta sheets. Like it's missing some of its secondary structure, which is sort of leaving, let's say, a gap or a groove in its structure, which is unlikely for the myophages because they have a very rigid structure, which helps them keep their straight tails shape. And it's normal in the sipho-phages to have that group, to sort of have the flexible tail. So JS1 had the sort of the comparison mostly like the flexible tail tube protein, where it had the groove in its tail tube due to its missing secondary structures.
Okay, cool. And how big was the phage genome when you guys sequenced the phage genome?
It's big. So it's not quite big enough to be called a jumbo phage, which many people do work with. And I don't remember the exact cutoff, but I am going to defer to Sabrina. But I think the cutoff is 200 and ours is 180 or something. So it gets pretty close to the size of a jumbo phage. It's certainly compared to the phage that we usually work on, it has very, very many more proteins, and in this case, a couple of tRNAs as well encoded in the genome.
Oh, wow. That's super cool. Oh, so tRNAs. Oh, wow. When I was looking, I was wondering, so no one's ever seen anything like this. Is this right structurally? No one's really seen anything like this, or?
It, you can say yes. So I think people have published on phage that, now, it's always easy to be smart in hindsight. Now, when we go back and read some of the previous papers, I think there were examples of phage that were that have turned out to be contractile phage, and the original data, normally just in a sub figure where, people easily lose track. It turned out that when Sabrina, read heavily to try and get to the grips with, why is it that it hasn't been seen before, that in fact, it may have been seen before, but just not recognized for what it was. So this is the first time I think that it's the first study, I think, where it's been put together, 2 and 2, that it's a contractile phage and also has a flexible tail tube.
Yeah. I was thinking, before we started today, this is why I love discovery biology. I mean, we're all mechanism, and that's fine. And it's great, right? But it's really fun to go out, right? And just look for things that maybe no one's seen, right? And then you figure out how they work, right? And I sometimes wonder, particularly like granting agencies and other things, I'm not sure they like that kind of work. They always tell, that it's not focused enough. But I think you can't find the cool stuff unless you go out and look for things, right? And sometimes you don't even know what you're looking for. It seems like kind of like this.
Yeah, absolutely. I would go, I completely agree. And I would say that, my lab actually is a discovery lab. So this is the sort of science we do whenever a project is conceived, even if the project is conceived by a student who's coming in thinking that she's going to work on Staph aureus and it's going to be all about, clinical interventions that we can learn something from a new strain or a new phage and get it into the clinic as soon as possible. I think there's really big scope for discovering things and for having labs that are funded to discover things. And then the people who are really good at translating things into the clinic, they should take over and translate things into the clinic. But I think you're right. I think if we, it's a little bit philosophical, but that's okay, I think, in a podcast. But, I think the more we lose sight of the fact that science is about discovery and it is about, working out what we don't know and then trying to work out, how to fill the knowledge gap. I feel like if we lose track of that, and certainly if we stop funding that sort of approach, then it stops being science, right? And it just starts being a sort of some version of engineering with, no new solutions that are coming into the thinking towards the translation.
Yeah, I 100% agree, and I sort of think, especially, I don't know, for you, Sabrina, it's kind of fun you discover something and you're like, you doubt yourself. Oh, is this real? Right. And all of these kinds of things. And then you realize that, and you go back and start looking, and other people had seen this before, but maybe not recognized maybe the novelty and sort of pursued that out. So I don't know. Do you think about that? I mean, what do you think about that?
Yeah. So I think it's really great to have the feeling of discovering something. Although, as you mentioned, that it has been probably seen before, but as Trevor said, that nobody recognized it like we did. And I think the credit goes to Trevor. Most of the credit goes to Trevor because he pursued the fact that we could actually recognize this and we could actually do the studies like measure the flexibility, do the structural analysis and stuff to make it really worthwhile that it is a new and novel discovery and it should be out there. So I think it feels really good.
Yeah, well, that's good. Yeah, because I think you're saying that a lot of it's Trevor. I mean, I think most people in my lab might say the same thing, but without the people actually going out and looking, right? I mean, that's the real discovery, right? That you go and you get excited about something you see. I mean, that sort of changes everything. And I would like to get back to this one point though that we talked about how this broad host range for this phage, right? This is to me really cool, and I was wondering what your thoughts are, Trevor, on this. And are there certain staphylococcal species that probably won't infect, I would assume, and then there are a bunch. Is there like similarities in cell wall depth or chemistry, or I mean, is there anything that you? Yeah,
we don't know. So just to fess up right at the top. So we don't know. We did think about this, obviously, and tried to work out, and in fact, that was the reason why the table of host range is, packed with so many species and strains, is because we were trying to look for a pattern. And I have to say, we don't know. It would be, I guess, a combination of two things. One could be about the cell wall, that the cell wall in some species or even some strains of a given species, it could be that there's some chemistry there that this phage can't get through, and that makes them resistant to JS1. That's one possibility. Or the other way around, it could be that there's chemistry in lots of the strains that is possible for JS1 to get through. But I feel like there's also the prospect that, one of the things we're learning more and more these days is that bacteria have all these anti-phage defenses, and those can be quite strain specific. And so it could be that the cell wall's not the issue, because we haven't addressed this. It could be that the cell wall's not the issue, but that there are some phage defense systems that are really good at stopping JS1 either from getting through the membrane or indeed from replicating once it's in the cytoplasm, and that those are strain specific. And perhaps a future student might want to go and have a look at the genomes of all of these things that are resistant to JS1 to look for a commonality that might be a defense system.
Yeah. I mean, one of the things that really, I think, been a great thing in microbiology, maybe the last 10 years or so, this resurgence of phage research, right? Like when I was in graduate school, you worked on phage if you were doing basic molecular biology, working out fundamental principles and really, great molecular biology. But now we've sort of moved back and we're sort of exploring phage in a way I think we haven't done in a long time. And Sabrina, you mentioned a little bit earlier about, how these could potentially be used for therapeutics. Can you sort of explain that a little bit more and about what the use and maybe what the use of your JS1 might be?
As Trevor mentioned, that we in our lab mostly look for the functionality, but if we think of the broad host range of JS1, it could basically infect some of the clinical strains which are isolated against some of the clinical disease as well, like some bronchiolitis and some other diseases as well. So if you think about the infectivity of JS1, probably it could be a good potential phage that could be used in the clinical settings to treat a lot of different Staphylococcus strains as well. And if we also think about the enzyme that we have, the peptidoglycan hydrolase enzyme that we have got from JS1, it also has very potent activity to lyse the peptidoglycan layers. So we know that for some of the cases, if the bacterial strain is resistant to the whole phage, we could use the enzyme itself as well. So I think in both the terms, JS1 as a whole phage can infect the cells as well, but at the same time, the enzyme that we extracted is very potent as well.
Yeah, I think there's a lot of really just amazing work coming out on phage therapies, right? And trying to understand, is this something that could be possible in the future? And I think, we all have heard about these really wonderful stories, right? About how, you've had folks with really antibiotic resistant, infections that have now been cured with phage therapy. I guess, sort of getting to the end, but I had a couple more questions. And one maybe, Trevor, is what does this do for phage classification? The fact that you kind of have this odd kind of, phage,
it's a really uncomfortable fit. So it's not as bad as it would have otherwise been, because it used to be that people working with bacteriophage relied on morphology as the classification system. So, in the olden days, not that long ago, people were still quite happy to think that any phage that's contractile should sit in this group with all the other contractile phages. Anyone that's got a long flexible tail is in this group with all the other long flexible tails, and anything that has a very, very small tail is over there in the group with other phage that have small tails. And it's really clear now from, excellent work done by the people who do the taxonomy of viruses that system just doesn't work. Like, it's a useful thing for people who are studying phage with a microscope, but it's not useful if you look at genome sequence characteristics. So the field has now moved to a point where we consider phage families and subfamilies and classify them based on genomes. And that then completely shakes up all of the morphologically based classifications. So from that point of view, a phage that is both contractile and flexible isn't such an awkward fit in the family tree, because it goes together with a whole lot of other phage that are also, we now know, we don't know how many of them there are, but we know there are now many of them, and they do all sit with, a sort of a branch of that tree based on genome sequence characteristics.
Okay. Yeah, cool. Well, Sabrina, I'm going to let you put your future hat on here, and I'm going to ask you, so what other cool morphologies do you think are out there for phage?
Do I think are out there? Well, certainly the one that I saw is the coolest to me at the moment. But I do think if we look for more, like we did in the wastewater, or if we look for more we tried actually to look for some other water sources which Trevor got from some distant parts of Australia. But yeah, so I think if we explore more and more of different environmental sources, environment has the coolest features in it. So I think if we explore more, we might see something new as well. But yeah, at the moment, I'd say JS1 has the coolest feature.
Yeah. Well, yeah. Well, yeah. Well, that's good. You got to like, I always tell folks in my lab, if you don't like what you work on, no one else will. Right. I mean, so you have to like it. So, in the last couple of minutes, I just wanted to ask you, Sabrina, and I bet I can guess what this answer is going to be, but what was your favorite part of this study, right? As a student, what was the time where you thought it's really cool to be a researcher and a student, working on this project? What was the result that was really cool?
As you guessed, and I think you guessed it right, I think it was, I don't have any other answer. I think it has to be this. I think it has to be this, when we figured out that it's something that we, as our lab, discovered in our lab, that this phage has a contractile and flexible tail, and not only contractile and flexible tail, but it has a range of flexibility. So I think I sort of liked that 2 or 3 months when Trevor told me to go through to analyze the flexibility, to analyze the curvature and so on. And I was trying to figure out which softwares to use, how to do that. I think I sort of liked that time of my PhD, which was fun. But at the same time, I was discovering something as well. So that time was really fun for me. And I think I was sort of proud of myself as a researcher as well.
Yeah, there's nothing better than seeing something cool when you come in the lab, right? When you first see it, right? It's it's amazing. And yeah, for you, Trevor. So what's next, right? What are you guys going to do next? What's the next set of experiments?
Yeah.
Other than make Suhani stay for another 7 years so she can keep working on it, right?
No, she will find better places to go, I'm sure. So what we will do, well, we are doing it now, so it will be the next thing coming out of the lab. We're part of so my lab is part of a centre of excellence here in Australia, and that includes collaborators who are physicists. And so I think you touched on this a little bit earlier, Marvin, but we are fascinated by the physics of this. So the idea that you can have a curved tube and that the internal tube can the external tube can contract across this curved tube, and thinking about the forces at play for both the contraction part, but also then for the DNA as it rushes down through this tube. So one possibility is that during the contraction event, the tail straightens and it's only ever straight when the DNA goes out. That's still a possibility. We don't exclude that at all. Another possibility is that it is curved and it stays curved, or it's perhaps flexible and can move, independently as the DNA is rushing down. And so we want to study that. So we have colleagues who will, write equations and do the things that physicists do, to have the theoretical part. And those equations will be informed by single particle analysis of the phage tail tube, so cryo-EM analysis that will give the parameters that go into the equations that then help us understand the mechanism. So at heart, as much as I love microbiology and I'm a professor in a microbiology department, I guess at heart I'm a molecular biologist, and I do like the idea of being able to, certainly to work with physicists or mathematicians who can really analyze the molecules and tell us things that we couldn't possibly have worked out ourselves. So I think that'll be the next thing. We want to really understand how this thing works.
Yeah, I mean, it's just a variant of a nano machine that, that we don't understand how it works. It's super cool, right?
Yeah.
So, so I think, this study kind of reminds us that even in areas of microbiology, at least to me, that we think we understand well, like bacteriophage, nature always has plenty of surprises, particularly if you're willing to go out and do some discovery-based research. So, I want to thank Sabrina Suhani and Trevor Lithgow, for joining us today, especially for getting up early in Australia while we're wrapping up the day here in Atlanta, and for publishing this work in InBio. I think this is exactly the kind of paper that we are really excited to be publishing in InBio. And I'd also like to thank you, the listeners, for tuning in to Editors in Conversation, the InBio edition. Remember, InBio is a journal of the American Society for Microbiology, which publishes InBio and also additional leading journals in microbiology. And if you join ASM, you receive discounts on publication fees across these journals. And if you enjoyed the episode, please subscribe, share it with colleagues. And until next time, I'm Marvin Whiteley, and this has been Conversations with Editors.